During prophase 1, the homologous pairs line up and exchange DNA through crossing over, which is what the caps are doing. During metaphase 1, the homologous pairs line up along the metaphase plate. The spindle fibers then attach to the centromeres, and that begins anaphase 1. During anaphase 1, the spindle fibers attached to the centromeres begin to pull them apart. During telophase 1, the homologous pairs have separated and a cleavage furrow begins to form in order to separate one cell into two. During prophase 2, the spindle fibers form and the chromosomes get ready for metaphase 2. During metaphase 2, the spindle fibers attach to the centromeres. During anaphase 2, the spindle fibers pull apart the chromosome pairs and bring them to opposite sides of the cell. The separated chromosomes are now on opposite sides of the cell, and a cleavage furrow begins to form the four gametes. During the era in Meiosis 2, the sex chromosomes don't separate while the rest of the chromosomes do. After fertilization, the male's XY chromosome combines with the female's X chromosome to form an offspring with an XXY chromosome.
The lined-up homologous pairs are crossed over and exchange DNA between the paternal-maternal chromosomes in prophase 1. This gives the gametes chromosomes that are unique to those individual gamete cells. Meiosis also allows for a combination of both sexes and genetic material preventing exact clones, unless identical twins are born, and increases genetic diversity.
If there is an error during meiosis, the produced offspring would have a mutation or defect. For example, an error in meiosis that affects chromosome 21 in humans causes Down syndrome. In less complex animals, it could cause a defect that can be fatal.